Our next speaker is Dr. Shahab Asgharzadeh. Did I say it correctly? Is the director of the Neuroblastoma Basic and Translational Program at Children's Hospital Los Angeles. He's an attending physician in the Hematology, Oncology, and Blood and Marrow Transplantation Department and an associate professor of pediatrics at Keck School of Medicine at USC. He went to medical school here at the University of Illinois in pediatrics and did his residency and fellowship at the University of Chicago. His lab at CHLA is working to identify the genetic differences in children who get tumors, neuroblastoma included, versus children who do not. His research is geared towards understanding the different outcomes of neuroblastoma, where the tumors have spread all over the body. His talk today will be on neuroblastoma biology and understanding molecular versus cellular therapies for neuroblastoma.
Thank you, Pat, and it's a pleasure to be here. It's my first attendance at this conference. And Pat emailed me and asked me to, because I think a parent's request, to discuss a paper. And I was like, oh, sure, I could talk about that paper. Then I read the paper. I actually had read it earlier, but it's a lot of differential equations and Bayesian statistics, but it's got a lot of biology. It's interesting and controversial. So I figured I'll start talking about that and go really basic at the first to describe some basic biology and genetics. So you have to bear with me, and for those who are biology aficionados, excuse that, the basic aspect of it. Before the disclosure, I'm the co-founder of a company called Simirex. It's interested in CAR T cells, especially for neuroblastoma, and I will not discuss the off-label use of therapeutics. But going back to the basics of biology. So this is going really basic, so I wanted to get you guys to the paper, because the paper is really complicated, but interesting. So as Dr. Goldsmith briefly mentioned, there's this concept of cells, and I'm talking about reverse back here, from gene, DNA, RNA to protein to cells to organs. And why I'm talking about this will become hopefully clear soon. All of us have different organs in our body that are made up of cells. These cells are very organized, and when you look at them under a microscope, they're very structured. And basically, when we diagnose a tumor, it's a similar idea, taking the tumor tissue, cutting it up, putting it in the glass slide, throw some color at it, and looking on a microscope and seeing whether there's disorganization or organization. This is an example of a liver tissue. Now, the liver tissue, each cell has made up of anatomy of it, which includes a nucleus and other organelles that have a certain function. So all our organs do a certain function, and the requirements for those functions are basically made up of these machineries called proteins. So what are proteins? Proteins are basically, in my opinion, you can think of them as Lego pieces. They're Legos that are functional, and they do a certain function. Each Lego is made up of these building blocks. There's 20 of them, 20 amino acids, or you can think of 20 different Lego pieces that make a whole different types of Lego structures that do a function. And every organ does a different thing slightly. So your liver is basically supposed to detoxify alcohol, clear up of our metabolites in our body, and it produces machineries, or these Lego pieces, that do that function. Versus a heart cell doesn't need those machineries. It makes different types of machines. But they're all abide by certain rules. They're all confined to their own structures, and they don't invade other tissues. This is really much different, and we're talking about different functionality of these organs. I want to point out the fact that there's about 20 to 25,000 of these Lego structures that could be made in any cell. But each cell at a given time uses about 8,000 of these proteins to do its function. And there are subset of classes of proteins, or these Lego structures, that are present in every cell. So they're used for dividing, for example, making an exact replica of the cell. So in your bone marrow, your blood cells frequently divide because they need to be produced new, versus your brain cells don't need that much division. So there's a lot of proteins that are involved in rebuilding that entire cell, the entire structure, over and over again. So these Lego sets are used for replicating. There's also, again, specific ones that do the specific function. Adrenal gland, which is the site of a lot of neuroblastomas arise from, are really for regulating your blood pressure, regulating some of your hormones as well. And moving on, again, bear with me for one more lesson in genetics, close to it, is going into how these proteins are made, right? It's actually a really beautiful way they're made. All the instruction sets for all those 25,000 structures of Legos that you make are made in a book, right? This book is basically built in a language that has only 4 characters. It's an English language. The 4 characters we call ACGT. And the book is about 3 billion characters long, and it's divided into chapters. The chapters we call chromosomes, and there are passages within these chapters that we call genes. And those genes, what we call genes, they're actually the building structure, they're the instruction set from how to make that Lego structure, the building block. And every 3 characters gives you specificity to one Lego piece. So when you read this thing, it says ATGATT, you've seen this thing in a lot of different things. Every 3 characters basically determines which Lego piece you put in front of the other one, and you build these things out, and your protein structure could be 100 pieces of Legos, or it could be 1,000 pieces of Legos. Whatever the function it produces, it's built that way. So the difference between each other is that every cell that we have has 2 copies of these books, 3 billion characters in length. One is from your mom and one is from your dad. And the differences in us is basically there's alternative spellings that occur in these passages that make the protein slightly do different functions, slightly, but that gives us our uniqueness because each cell has a copy of it. And they only have 2 copies of the book. And each book has 23 chapters in it. It's duplicated, 46 chromosomes total. And when there is a mutation, what we call a mutation, as you guys think about an ALK mutation, the mutation is nothing but a bad spelling. It's basically an error in the spelling of that instruction set for making that protein. So the mutation would basically lead to a protein that has a different function or loses its function completely. So when we talk about ALK mutation, when we talk about MICA amplification, it's basically that passage, which is in chapter 2, verse part 1, is basically under a Xerox copy machine and replicated multiple times, sometimes 20 times, sometimes 50 times in a cell. But again, remember that each cell has only 2 copies of the book. Now, what happens with cancer? Cancer is, you could think of it as a dysregulated organ. So you basically have now an organ in the body. The body recognizes it as an organ, doesn't think of it as a foreign initially, and we'll talk about that why. But it doesn't abide by the rules of this book anymore. It deletes parts of the passages, it deletes chapters, it Xerox copies other passages, or Xerox copies the entire chapters, and it no longer has what equivalent of the 2 book. It has different chapters missing or chapters completely replicated, as this sort of image shows. And it makes certain proteins more or less, and it invades other organs. And it also produces certain proteins that dampen your response to that organ that's dysregulated. So that's why it's actually formed, because the body doesn't recognize it, recognizes itself again. So the T cells that are supposed to basically go and actually kill this tumor are no longer doing their job. Okay? So in neuroblastoma, basically the normal adrenal gland should have 2 books, the equivalent of 6 billion characters. It should have 2 chapter 1s, 1 in each book, chapter 2 all the way to chapter 23. In a lower risk neuroblastoma, what typically happens is the amount of characters that are present is about more than 2 books. It could be 3 books, could be 4 books, sometimes 5 books worth of characters. And there are whole chapters that are completely duplicated or chapters that are completely lost. So we call these chromosomal duplications or chromosomal deletions. Just think about it again as chapters. Versus in the higher risk neuroblastoma, there's about the same number of pages or characters in 2 books, but what's happened here is there's regions of the chromosomes that are deleted or amplified, duplicated. Parts of the chromosomes, parts of the chapters are missing, part of them are gained. And this basically concept is sort of like what we look at when we actually get a tumor out, and we could analyze this and give you an idea of whether you have what we call segmental chromosomal aberrations, which is a fancy word for saying you have segments of your chromosomes missing or deleted or duplicated. Another concept I want to go back to in a second, or right now actually, is for each chapter, the book is a little bit unique because at the beginning and the end of the chapter, there's bindings that are very strong, and these bindings maintain the integrity of that chapter. And those bindings are basically in biology called telomeres. So the tumor cells really try to maintain those bindings so those chapters are intact, because otherwise it will die. So what does this actually look like in real life? You have a low-risk tumor on the left-hand side. The 3N means that there's about triplicates of the copies of the book. Those are chromosomes, or those are the chapters that I'm mentioning, and we actually could look at them, and we could count them, and see there's 3 of them or 4 of them, so that we know which chapters are gained or which chapters are actually completely deleted. In the middle, the other 2 sections are about 2 ends. Those are basically showing that there's chapters that part of them are deleted and part of them are gained. Sometimes a chapter goes and binds to another chapter at the end of it. So there's one section, let me see if I could, oh yeah. So that one, the chromosome 17q is basically added. And then on the right-hand side, you see the same chapters, but you see these tiny dots there. Those tiny dots are actually MECN DNA that are so much replicated on a Xerox machine that in the cell you could actually see them. They're actually present, and they're giving the instruction set for the cell to make the MECN protein. All right. So another interesting thing about neuroblastoma is that there are repeating themes of chapters being deleted or gained across neuroblastoma. So 17q, for example, is often seen amplified or duplicated in 2 copy numbers in neuroblastoma cells. So 2 more slides about basics of genomics. So going back to the mutations, there's one other concept I want to talk about, which I talked about a little bit, is this variance of alternative spellings. So the variants are basically spelling changes that cause minor or no change in the function of the protein that occur in the book as well. And you guys also will hear about epigenetics and epigenetic drugs. All that is basically is that the spellings aren't changed, but there's 2 highlighters. One that's like a yellow highlight, that the cells basically go across a passage and means that like really explain that or produce that protein more without actually duplicating it. Or you could have like a black marker, which also you could go across and highlight, and that says do not produce that protein. So it's an alternative way of regulating the amount of the instruction sets for that particular machinery or protein that you want to make. So in neuroblastoma, there is mutations that have a really large effect size, so ALK and FOX2B, that like gives rise to the tumor driving it. And there are these mutations that have a lower effect size, means that you have to have multiple copy of them. Even the alternative spellings that actually have to have multiple copy of them to give rise to a particular tumor. And this is sort of shown that the names of them. And again, a mutation is something that when you change the instruction set, it leads to either a loss of function, so you lose the wings of the plane, they can't fly anymore, or you can have alternative spellings where you give maybe different markings or maybe a stronger engine, and that's what basically occurs. So, key things is that neuroblastoma is a complex dysregulated organ. Each cell now, remember, has its own books and passages with, again, repeating themes of certain chromosomes being deleted. Not each cell in a tumor has the same chromosomal or chapter changes or passage changes. So not every cell has an ALK mutation, but a majority of them do. Tumor cells can, when they replicate themselves, they can introduce new alterations, new spellings as well into the tumor. And the tumor growth basically is equivalent of evading your own immune system. And there's few tumor cells within here that act as progenitors. So there's a concept that's coming into neuroblastoma as there are phenotypes in neuroblastoma where we call mesenchymal versus adrenergic. A majority of the tumor cells are adrenergic, but these mesenchymal cells, you can think of them as sort of like progenitors, that when you give the chemotherapy, you kill a lot of these adrenergic types, but we think these mesenchymal types of neuroblastoma stick around the body and give rise to more adrenergic or their progenies as well. And when we look at a tumor under a microscope, it's complex. It's got endothelial cells which deliver oxygen through red blood cells. There has other immune cells that are now co-opted to help the tumor grow. It has support cells to help the structure of the tumor. But all of this sorry for the first 10 minutes, was to get you guys to a couple of other places. So last point, just to tie up this basic biology, is when you think of chemotherapy, chemotherapy is nonspecific, and it basically goes after the proteins that are involved in replicating the cell. So anything that divides fast, basically, you guys already know this stuff, is what chemotherapy attacks those particular proteins. Versus a targeted therapy that Dr. Goldsmith talked about is basically aberrant proteins, the proteins that are produced abnormally high, or that has changed, that we could actually target now those proteins because they're only specifically highly produced in tumor cells or aberrantly produced with this alternative function or alternative Lego structure that we could attack that modified structure. So this was the paper that Pat said, Shahab, can you talk about this? So it said, neuroblastoma arises in early fetal development. Its evolutionary duration predicts outcome. And there's a couple of the figures that the paper they're talking about. And I thought about giving a differential equation talk as well, but I decided against that. But the genetics was important to get you guys to the next topic. So, incidence of neuroblastoma. It turns out that at birth, the first year of life, the incidence is about 70 children per million children. And you may think that's really high, which it is, but we don't actually diagnose a lot of these children because we used to. In the 80s and 90s, we actually did a screening test where we took the urine from infants at 6 months and 9 months and tried to see whether they have telltales of neuroblastoma in their body. And there were a lot of children that we identified. That's why the incidence is so high. But it turns out most of those children had these neuroblastoma that are very low risk and they go away on their own, basically. We never see it. Japan tried to take them out, and they did. But what happened is we found out that all of that work basically did not decrease the number of children that would have a higher risk tumor later on at 2 years, 3 years, 4 years of age. So that practice was stopped. But the incidence is high in the first year of life. And typically, the kids that are less than 1.5, 2 years old, except for those that have a MICAN amplification, have a low-risk type of tumor, and the ones that are older have a higher risk of tumor. So I mentioned the screening infants did not identify children that were high risk. And what also is really interesting, when we look at the protein content, again, so what type of proteins are produced in neuroblastoma cells, it's really similar to the proteins that are produced in early neuroblast cells that are seen in the first 8 weeks of gestation. So the question becomes, is neuroblastoma cancer of adrenal gland cells in older children that de-evolves, means goes back in time, and produces these proteins that are like, resembles the early neuroblast, or is it really an embryonic cell that's formed really early in pregnancy and just sits there for a long time quiescent before it actually develops into a neuroblastoma? Or do we have these cells all in our body in infancy that are waiting to be hit by something and become neuroblastoma, basically, right? So those are the questions that we really don't know the answers to. And this paper tries to look at that. So this is a graph from the figure. Now you guys can really understand what these are. I'm going to tell you what the columns are. The columns are basically tumor samples that they looked at. Those on the upper part, the chapters, basically. So every dot that you see here, the black dots, represents whether the copy number of that chapter for that tumor is gained. So it says 17Q gain. Q and P basically are beginning or the end of the chapter. So basically the 17Q means the end of the chapter is gained. 17 gain means the entire chapter is gained. So there's a lot of variability, but as you could see, there's some common themes. So about 96% of the samples have traditional chapters that are gained or lost within this sequence. So 17Q, 1Q gain, 2P, 2 gain as well. We see a lot of these repeating themes of the chapters being lost or gained in neuroblastoma. At the bottom part is these passages of the genes that I mentioned, and there are changes in their spellings, basically, and there could be different types of spellings that could occur in different parts of the passages, but those are the mutations that occur, and I won't worry about the alterations, but that's basically synonymous to whether you're reading the entire protein and part of it gets deleted because of the alternative spelling or what happens. But there's, again, a little bit more of a paucity of the mutations that occur in the bottom part, but they're still there. And then the last part that says TMM all the way at the bottom, basically that's the telomere maintenance machinery, and that's, or mechanism, that's the binding that I told you. So that these cancers, if they're really good at maintaining their bindings for the chapters, it's denoted as red, basically there. And then there's also stage 1, 2, 4S, stage 3, and stage 4 disease all the way on top. So what they try to do is they look at this, this is one particular tumor, and the diagram that you see basically, that you see black dots across and or green dots all the way on top, this again goes the same idea. There's different chapters for a particular tumor, and for example in here, the chapter 1, the latter part of the chapter is gained in that green part, versus there's parts of chapters that are normal in copy number 1, and then other ones are deleted or lost. But we could actually look at each part of these chapters and measure the alternative spellings that occurs in these things to get an idea of how old this book is. And what they actually found very interestingly found that among the cells, you could tell that there's alternative spellings that are across all the samples, meaning that they originated from a really early progenitor cell. And then there's somehow early on, there are changes that occurred in some of the chapters that gave rise to neuroblastoma. And that's basically showing this 2 clonal copies. There's 2 copies of this that has an increased frequency of these alterations. And that's basically denoted down here as how this evolution occurs. But they're basically saying that there's 2 types of neuroblastoma progenitors that you could see. There's one that is a early common ancestor and then the most recent common ancestors. And they go on to nicely show, that in neuroblastomas that you could see, there's types of neuroblastomas that have this early, most recent common ancestor cells, and there's types of tumors that have both this early and the most recent, and there's few of them that actually have no early common ancestor. What that translated into actually is survival as well. The tumors that had just the early, most common recent ancestor have the ones that didn't have this binding capability of keeping the chapters, so they didn't have the TMM, the maintenance of the telomeres, and they had an excellent outcome. Versus the ones that had both the early common ancestor or most common recent ancestor had actually worse prognosis. And there were ones that had the 17q gains and this ability to maintain their chapters really well. So then they did some fancy mathematical modeling to basically show that if you think of this as the early common ancestor occurring, staying quiescent, and then gaining some abnormalities later on in life, what would be based on the incidence of 1 in basically 100,000 children, and then computing the number of mutations that could occur and how often these cells turn over, and the oncogenic events, they estimate that this early common ancestor occurs in the late first trimester of pregnancy. So there's something that happens and not all of those become high risk, right? Because a lot of them do become basically these, what we think these neuroblastomas in situ, and they go away. But this is the first telltale sign. So this definitely requires further validation. And we could think about looking at even single cell fetal tissue, aborted fetal tissue, to look at this thing to see whether we could see these alterations in these single cell analyses. And they're doing a large study in Germany on additional tumors to validate this finding. But this is sort of the first clue that we're getting that maybe some of these early events that occur really early on. Why they occur, we do not know. All right, so I'm going to change a little bit gear towards what I actually spend a lot of my time on, which is inflammation, cancer immunotherapy, and trying to figure it out. I'll give you guys a little bit more of a introduction to what I call a tumor microenvironment as well. So in order for the cancer to actually evolve and get larger, it needs to evade our immune system. We, as we get older, have cells that become tumorigenic all the time. We don't realize it, and our immune systems actually take care of it without us knowing. That's why a majority of the people who get lymphomas or other cancers are the elderly, because our immune system gets weaker. So once the tumor occurs, a small tumor, just few cells, what happens is that it goes back and forth, the immune cells comes in, the NK cells come in, and they kill it because the tumors are producing these abnormal proteins, and they recognize and they're killed. And there's some things also intrinsic. If they don't have their maintenance, the telomere maintenance ability, they also devolve and die off. And we think that's one of the reasons why that the stage 4S tumors as well, or these neuroblastoma in situs, go away, is because they don't have that ability to maintain their telomeres. So at some point, again, this occurs, the tumors get eliminated through either the NK cells. There's also this dormancy, we believe, that occurs, and there's a lot of evidence that shows that this does occur, where the tumor remains dormant and it's like I will not grow and the immune system will not kill me, I'll stay there, until an event occurs again that then the tumor cells get the ability to hide away from the immune system. The immune system no longer is able to keep it at bay, and they produce different proteins that, again, shut down the immune system. One of those proteins is called TGF-beta, arginine that Dr. Goldsmith talked about as well. All of those things come into play when the tumors escape the immune system. So our interest, my interest in the lab many years ago, was to figure out why children who have a metastatic disease that are less than 18 months of age do so well with just few cycles of chemotherapy versus the kids who are older than 18 months for MYC non-amplified subtype do so poorly despite giving chemotherapy and transplant. And one reason is basically they do have a different genomics. So again, the younger ones have the whole chromosomes, whole chapters amplified or deleted, versus the younger ones have this abnormality in terms of the tumors have this abnormality in terms of deleting certain passages as well. So we looked at a lot of these tumors, and what we found out basically is that again, it's a fancy figure, but each row of that upper, what we call a heat map, denotes the expression of the level of the Legos or proteins that are made in those tumors. Each column is a tumor. And then they're sorted by age at diagnosis. These are all metastatic, children with metastatic neuroblastoma. And as you can see, right after around 18 months of age, there's a lot more red on the right-hand side than there's on the left-hand side. And these are proteins that basically look to be related to inflammation. So somehow the tumor gets inflamed, lots of immune cells come in. And when we look at a microscope, we actually saw that the tumors that come into the tumor, the immune cells that come into the tumor are these cells that we call macrophages. And they play a big role in supporting the immune cells I'll talk about in a second. But we actually built a predictor to figure out for the kids that are older than 18 months whether this inflammation, along with some other tumor markers, denotes a worse prognosis or a better prognosis for certain patients. And we developed this high-risk, low-risk category for the patients. Going back to the macrophages, these are immune cells that are always in our blood, and they have two functions. One is to they become into macrophages, and the macrophages either engulf bacteria and have anti-tumor effects, or there's these M2 macrophages. When you cut yourself or you get injured, these macrophages come in and try to heal your body. So the macrophages that come into the tumor are similar to the M2 macrophages. They're actually trying to help you. So whenever you get chemotherapy and you damage a neuroblastoma, what next happens is that a lot of monocytes come in and try to actually heal the tumor. The body doesn't, again, recognize this as a foreign part of the body. So a lot of our efforts are into figuring out how to actually block the effects of our immune system, helping the tumor in these cases. We go a little bit more quickly through this, but we have a mouse model of neuroblastoma, and we actually see this as the neuroblastoma develops. This does happen. The macrophages in brown, these are all cells that on top, and the top is just a normal adrenal gland, and the bottom one is a neuroblastoma that's developing. You see all those brown spots are macrophages that come in, and when you put macrophages together with the tumor cells, they actually grow faster. So long story short, this basically interaction between macrophages and neuroblastomas has a lot of communication. The tumor cells tell the monocytes, hey, come into my area. They produce a lot of proteins, including TGF-beta, and that leads to the growth of the tumor cells through amplification or increase in regulation of the MYC. We've also recently found out that there's other cells that supporting structures called fibroblasts together with neuroblastomas, also produce a lot of this TGF-beta that we think actually promotes not only the tumor growth and evasion, but we're also seeing that they actually help maintain some of those early progenitors that I mentioned to you, the mesenchymal type of tumor cells. So after chemotherapy, when the macrophages come in, they're actually helping regenerate or induce the expression of proteins that are of these neuroblastomas that give rise to more neuroblastoma. And the TGF-beta itself keeps the T cells away, and including CAR T cells. So this is really a major issue. So a complex figure with a tumor cell, NK cells, CAR T cells, these MDS or macrophages are what I'm talking about. They're all trying to get rid of, push the T cells out of the way and producing these various proteins. I'll talk a little bit about CAR T cells now, briefly, just to give you the challenges that we have in CAR T cells. I know you heard it from Dr. Hexey yesterday and Dr. Goldsmith a little bit. I'm going to give you a little bit just a big principles on how we are trying to tackle this thing. CAR T cells, as you guys know, it's a regular T cell that you get out of the blood. You again engineer that DNA to make a chimeric. Chimeric is basically a half of two different animals, and we're making new Lego pieces, right, that we put on top of the T cells. And the purpose of this new built machinery, a new protein, is to be able to go and bind to the tumor cell and kill it. We grow these things in billions outside the body, and we give basically 500 million to 1 billion cells back to the patient. And it works beautifully in leukemia. And the reason it works really nicely in leukemia is because as you infuse these CAR T cells back to the blood, the first thing that they see is leukemia cells running around the blood, because that's a blood cancer. But you could think of neuroblastoma and a solid tumor. What happens, these solid tumors produce that TGF-beta that I told you as a cooperation with macrophages. And the TGF-beta basically disables the T cells. And also when you inject 500 million or 1 billion cells into a patient, they have to find their way into the tumor and then set up shop and grow and try to kill the tumor. That's a very hostile territory they're going into. And there's not enough CAR T cells. So one of the main challenges in the CAR T cell is how do we create a more hospitable area for the CAR T cells to do their job? And this is a research that I'm focusing on, and hopefully in a year or so I can tell you guys that we're starting a clinical trial on a new chimeric that we've developed to help offset the effects of the TME with CAR T cells. The other challenge in CAR T cells is off-tumor toxicity. So some of these proteins that we're going after, GD2, as you guys remember, are also present in the normal nervous system and also in our central nervous system tumors. So we did an experiment where we tried to make a really, really good GD2 CAR T cell for a mouse neuroblastoma. Highly effective. And then one that, actually we didn't know, and one that was not as effective. So basically these mice on the left-hand side, they continued to develop the tumor, and one of them got cured, but a lot of them died due to the tumor, versus the really good CAR actually infiltrated into the CNS, into the brain of the mice, and actually killed the mice. So we don't want that as well. So we have to figure out ways to make these CAR T cells also safe. And one way we thought about this thing is to have, again, a complex figure, but having a dual approach. So basically you could do this as a dual CAR T cells. You could attack two antigens or two proteins on the tumor cells. Or you could have this what's called a gated CAR T cell, where you basically send a sonar, and the sonar says, yes, there's a tumor nearby, get the torpedo ready. And that's the concept basically that we went after. So you have to have both of the markers on the tumors before it kills the tumor. And we were very successful in this approach, and this is another approach that we're taking as well, where we have these B7-H3 CAR T cells that cure the tumor, but we wanted to make this gated approach, and again, it's a fancy picture for that same thing, a sonar technology that actually activates, when you see the GD2 on a neuroblastoma, it activates the B7-H3, and it primes it to actually bring the B7-H3 out to kill the neuroblastoma tumor cell. And this approach has worked really well for us, again, in our animal models, showing that we could actually go ahead and create these B7-H3 GD2 or GD2 B7-H3 CAR T cells And even though some of the CAR T cells actually may get into the brain, animals do not die because the sonar is shut off back then and the CAR T cell stops functioning. So these are the two challenges that I mentioned, and my lab is very active in this pursuit. So in conclusions, basically advances in single-cell technologies that where we go and really read the data from all these cells will help us identify the origins of neuroblastoma tumors. Understanding the tumor microenvironment, the macrophages, the fibroblasts, how they interact with the tumor cells are really important to think about new opportunities to improve our combination therapies with immunotherapies. And the CAR T cells are really exciting to me, and hopefully in a year or so I could come back here and tell you guys about a clinical trial that we're going to start with these CAR T cells. But there are definitely challenges. Again, thank you for all the participation in the clinical trials, and a lot of the people who've helped me, including family foundations, to get my career going initially and help me continue to do the research in neuroblastoma.
Thank you.
Take any questions.
Does anyone have any questions? No, you got them this time. Oh, okay. Kate's got one. We've got one here.
I'm so fascinated. Thank you so much for explaining that as chapters of a book. That was really helpful. And it's just a really people-friendly way to understand genetics. So thank you. I wanted to start with that. But I wanted to ask, when you're examining these genetic sequences and you're coming upon these genes that we know are affected by neuroblastoma, first of all, are you able to kind of see which book this comes from, right? There's mom's book and dad's book, and does that genetic code play any, does that help at all to examine genetic sequences or family? I know neuroblastoma is not like a family history disease, but does it help in the research to identify, and are there related conditions on the same genes, things like that help?
You mean in terms of how the neuroblastoma was formed initially?
Yeah, or does that make a difference? Does it?
It
seem
does.
to
I
matter?
mean, there's a long. I come back from I got my interest in science looking at Lou Gehrig's disease and ALS, and there was a familial cause of ALS, and we looked at a lot of genes and be able to figure out why it caused Lou Gehrig's disease. So John Maris, and I helped slightly in this project as well, long ago, we tried to look at a lot of neuroblastomas and tried to look at a lot of the genes to see whether the normal genetic of a child predisposes them to neuroblastoma. And what we found is there are certain genes that may predispose, but the effect size is so small. And by effect size, I mean, you take cigarette smoking, right? If you smoke a pack of cigarette for 10 years, you have 8 to 10-fold higher risk in the population, actually sometimes more, 20-fold, to get a lung cancer, right? When we see these genetic alterations, these alternative spellings in children's blood that we say predisposes to neuroblastoma, the incidence, the risk of developing neuroblastoma, it's like 1.5 times higher than population. So we can't for certainly say that the child will develop, but it does give us an insight into the biology of neuroblastoma. So that paper basically says that there is something that happens, right? The second trigger is unknown still, and it may just go its course and evolve and then get smaller again and regress in the first year of life, which majority of neuroblastomas do. But what keeps that second hit and when that occurs is unknown.
Okay, one question back there.
Hi. Yes, thank you for kind of putting a lot of this in layman's terms, right? Like the explanation of some of those challenges with CAR T with solid tumors versus a blood cancer like leukemia. If a patient goes into a CAR T with a lot of marrow disease, would that, I mean, obviously we have the challenges with solid tumors, those clusters of cells, things like that. But would marrow disease with neuroblastoma would you expect a similar outcome like as a leukemia that's like those free-floating cells? Would that, I guess, how does that correlate? Is that?
Good question. So it will be a harder challenge in bulky tumors in general, right? But if you're talking about if you have a child that's relapsed that has smaller bone marrow metastases or bone metastases all over, which is typical what we see. We don't see a giant bulky tumor because we do a lot of testing, CT scans, MRI scans, MIBG scans. I think it does give you an advantage over a bulky disease. In animals, if you have a large bulky disease, especially for neuroblastoma, interestingly, compared to other solid tumors, there's a lot of interaction that causes, as we discussed a little bit yesterday, some CRS syndromes, even in mice. But the challenges with the TME remains the same. So in the bone marrow still, there is a microenvironment niche that helps protect the immune cells, tumor cells from the immune cells. So you still have to overcome that barrier. You may have a little bit of a less of a challenge in terms of the circulation and getting to that place compared to actually a bulky tumor. But the TME challenge still remains the same. But we have to test it. That's what the goal is.
Right. Thank you so much.
Certainly.
Okay, another question?
Hello. In regards to the T-cell approach, I understand the challenges with T-cells has been persistence and expansion. Are you in a position right now to disclose is that still a challenge? Has Rome overcome that?
Yeah, very good question. Again, you think about number games. The reason we give chemotherapy in this called lymphodepletion chemotherapy before every CAR T cell is because typically in our body there's billions of cells, or 50 billion white blood cells, to 200 billion white blood cells running around our body. And reason we give lymphodepletion is to decrease this number to allow the expansion of the CAR T cells that we give the patients. And the amount of CAR T cells we give the patients, typically around 500 million to 1 billion cells, right? Some with billions probably a little bit higher end. And to allow them to expand. So it turns out that for most of the CAR T cells that we have, they expand up to about 100 cells per microliter for around 2 weeks, and then they go down. Mm-hmm. And that's a big issue in terms of expansion. So the way we're approaching it with the construct that I didn't really go into detail is we're using TGF-beta that the tumors secrete as a way to also expand our cells in the blood. So that the CAR T cells that we're constructing, I'm happy to disclose this, basically uses that TGF-beta and reverses it in the T cells, allows them to expand, which the TGF-beta actually in a lot of the patients with neuroblastoma relapse is higher than normal individuals. So we could expand it, and then once they get to the tumor, there's a lot of TGF-beta as well there, and that will aid them in doing their job, which is killing. So expansion is a big issue. You don't want it to be huge, because then you'll cause problems as well, the CRS and a lot of other things, and off-target toxicity. So we have to figure out the therapeutic range that we want to do this.
And persistence?
So the persistence, again, is for me, is also tied to the using the tumor microenvironment. If you eliminate the tumor, you eliminate the proteins that are secreted that maintain that environment, then your CAR T cells will get lower in number. But for all these CAR T cells, there's importance of also putting in a suicide gene, basically, that you could activate in case there's problems.
Thank you.
Sure.
Hey, anyone else? Any other questions? Okay. Thank you.
Of course.
We're going to go ahead and take a 15-minute break, so be back here by 11:05.
Yeah, no, I'll be. Yeah, but, and my slides will be, my slides won't say anything, but they'll show up here.
Yeah.
Cool.
Yeah.
Thank you.